Thank you so much for that introduction. Can everyone hear me okay? Wonderful. And the slides are full screen for you all? Wonderful. So thank you so much. And I really appreciate the shout out to community college and also the interdisciplinary background. Yeah, I think many times we're trying to put ourselves in boxes and we don't always need to. And I think that's an exciting new way academia is actually transitioning to. So that being said, today I'm going to talk to you about cancer. And you think, oh, anthropology and cancer, what's the intersection? I want to study the evolutionary vulnerabilities to cancer, specifically why we're vulnerable to cancer, why humans are vulnerable to cancer. And to actually ask that, I look at everything but humans and look across the tree of life and see the consistencies, the parallels to human biology, and then some of the differences. Um, I'll talk to you about, again, the phylogenetic aspects of cancer biology, and then I'll end the talk talking about within humans. I can't do this talk without mentioning the team that I'm a part of. I work with the Arizona Cancer Evolution Center. We call ourselves the ACE team. And I feel like it's a dream network of colleagues. We have evolutionary biologists and cancer biologists, and we have veterinarians and math modelers and oncologists, and we have patient advocates. And it's a really big interdisciplinary team. I feel like I joined the Avengers in some aspects of it because we all have our own special strengths and superheroes. And I want to say some of the most important contributors to our team are the patient advocates and telling us their insights as they go through their cancer treatments as well. And I can talk more on that in the end because we do a lot of outreach with them. Okay, so starting from the beginning to talk essentially about cancer, I wanted to give some first principles. And cancer is basically a disease of somatic evolution and mutation. Every time a cell divides, it has a chance of mutating. This is just essential to cell division. There can be mistakes that happen leading to a mutation. And so then there's decisions that need to be made within the cell when it has a mutation. It can fix the mutation. It can kill itself, apoptosis. We'll talk about this a little bit at the end. That's a really good way of defending against cancer, is just apoptosing or cell death, because then you can't pass on the mutation. And sometimes cell repair is not perfect and mutations can happen. The other option is in the cell with the mutation. Maybe it's in a key gene that does proofreading and suddenly it can't notice its own mutation, or sometimes they just slip through. And then you see this accumulation of mutations over the life course, eventually leading to cancer. And this is really inherently important when we're thinking about the fundamentals of cancer biology, is this cell division that happens. And briefly, in the past few years, we've started mapping cancer across the tree of life, and we find it's ubiquitous in nature, especially with multicellular organisms. It's come hand in hand. As a cell starts cooperating, or as an organism's cells start cooperating, there's suppression of cancer. You have cancer suppressant mechanisms that must have co-evolved. And so, like, for example, you see this beetle right here, and it has a tumor. Some would argue whether this cacti right here with these fasciations is indeed cancer, but it is over proliferation of some tissue. It's interesting, as we've surveyed cancer across the tree of life, some of the weirder forms of biology, fungi or hydra, it's very hard to define cancer. And we've realized that cancer is defined very anthropomorphically. And as we've tried to expand the definition of cancer, we've hit many roadblocks because there's just not the same tissue structure in other organisms. But we would argue that cancer has been a persistent selective pressure in multicellular organisms for the past 3 billion years. But if this is the case, and cancer is ubiquitous, or cancer has been a strong selective pressure, why haven't all species just evolved amazing cancer defense mechanisms? Why have they not just solved the problem of cancer if it's this evolutionary ancient problem? And also, as we look across the tree of life, why are some species better at suppressing against cancer than others? And to answer this, we went into an evolutionary framework. Before I get into that framework, I just wanted to give you a few more definitions for individuals that are studying cancer. I'm going to talk about neoplasms, and that's just a general term for abnormal growth. It's benign and malignant tumors included. Malignancy is cancer. So a malignant tumor, these malignant cells actually invade the nearby tissue. They break through and invade. So that's the cancer definition. So looking to evolution to help us answer some of these questions. Why do organisms still get cancer? And I think one essential feature of explanation is that there's just trade-offs. So there could be a trait that increases an organism's reproductive success, and as a side effect, increases that organism's chance of cancer later in life. This is also a term called antagonistic pleiotropy, where there's just two effects. And if one of these effects is beneficial early in life and selected for, then maybe the cancer phenotype can still arise. An example of this you can think about, and our team likes to think about selection for growth, getting big. And again, the cell number problem that I talked about earlier. So bigger organisms are less susceptible to predation, and this may lead them to have greater reproductive success than their small rivals. So there could be an evolutionary force to get big. However, having more cells requires more cell divisions. And these more cell divisions can increase the chance of that bad mutation that leads to cancer. Okay, so it's risky to have more cells. Then also rapid growth. We think about it in terms of not only the quantity of cells, but how quickly cells are dividing. And if the cells are dividing quickly, maybe you don't have as good of a job as proofreading and repairing this DNA damage. But again, if these features, as you know, rapid growth or getting bigger, enhance species reproductive success, that just might be the trade-off that is compromised. And we see a few examples of this in the literature. I apologize, there is going to be an image of a tumor that comes up on a deer right here. This is an antleroma. And of course, antlers are quite important for the male species to do reproductive competition. They're the most rapidly growing appendage in mammals. They grow very quickly. So we could maybe predict that even though these species have these appendages that grow really quickly, the trade-off is they might be vulnerable to certain diseases such as cancer. Another example is melanoma. So in this fish in the bottom image right there, that black spot on its tail is actually melanoma. The interesting thing about this species, this is a male species, and the females preferentially mate with the largest male. They actually have a mutation in a growth factor gene that makes them bigger than other species. But later in life, they go on to develop melanoma. But this mutation stays in pretty high prevalence in this population because the females preferentially mate with the individuals that have this mutation. Okay, so this way we call antagonistic pleiotropy, but again explains why we still don't see complete cancer defenses across the tree of life, because we're selecting for other things like growth. Another reason why we might see cancer across the tree of life, and this really hits home when we're talking about humans being vulnerable to the disease, is we may be in something called evolutionary mismatch, where we're just in a novel environment. So your original environment is now much different than the environment you're currently in, and that there's an adaptive lag. There's a lag in potential cancer defense mechanisms. In these novel environments, of course, it could be from like savanna to cities, right? But also the novel environment could be viruses, new population densities, new pollutions or toxins that we're currently not adapted to. And one component that I think is really important to point out, and especially when I'm talking to a more medical-based field, as you that are anthropologists might be familiar with this weird acronym that is Western educated, industrialized, rich, and democratic, but some of the physicians that I talk to actually are unfamiliar with it. But the weird populations only make up 10 to 15% of the world, but a lot of our research focuses on weird populations. And we suggest some weird populations might be an evolutionary mismatch. One example of this is Mel Greaves out of the UK has recently proposed that certain childhood leukemias may be due to an environmental mismatch. This is linking actually the hygiene hypothesis, this lack of exposure in certain pathogens in weird populations. So the incidence of this cancer has increased substantially in Western societies. And so he's predicting that our immune system is actually not calibrated properly due to this pathogen, lack of pathogen diversity. And then it is then triggered by an abnormal immune response to a common infection and can lead to a cancer phenotype. So this is just one example of providing a potential evolutionary mismatch scenario in why humans would be vulnerable to certain cancers. I will end this talk specifically talking about breast cancer in this context. I do want to mention, though, that cancer has been with us since the dawn of multicellularity, and past populations of humans likely dealt with cancer as well. Maybe not as much, or maybe different kinds of cancer, but it definitely wasn't a zero. And so I worked in collaboration with a paleopathologist, Karina Marks, and we just asked, this was a literature review and some of her own data, and we were looking at temporal trends. Did we see cancer start increasing, especially since the onset of agriculture? And we actually didn't find any temporal trend in the fossil record in past populations. Obviously, this is a limited data set looking at fossilized records, but we concluded either the mismatch was really recently in our human history, or maybe cancer was always a problem, but it's just shifted. Maybe cancers that were derived from infectious diseases used to be more of a problem in past populations, and now it could be more cancer related to diet and activity levels and environmental carcinogens like smoking. And so obviously there needs to be follow-up on this, but current data suggests that there isn't this temporal trend in past populations. The last thing I want to focus on when I'm talking about evolutionary perspective to get cancer is just looking at life history trade-offs. And this is how I'll focus the rest of my talk. And so what do I mean by life history trade-offs? Well, according to life history theory, which many of you should be familiar with, resources are finite. And so then organisms are subject to trade-offs. Our three praxis of trade-offs that we think about are reproduction and growth and somatic maintenance. And then if an organism invests heavily in reproduction, then it has to come from some part of this resource pie. Maybe they have less investment in somatic maintenance. And what we can see across, the tree of life is species have different life history traits guided by their ecology. And so you can have organisms that have invest heavily in reproduction, low survival, have many offspring. And this is a continuum to the other end of the axis where you have elephants, which are slow life history strategists, They invest heavily in their somatic maintenance to get big and long-lived, and invest heavily in one offspring at a time. So they end up with few offspring over their life period. And what we want to point out is within this somatic maintenance category is cancer defenses, essentially. So that's DNA repair, cell cycle control, and immune function. These are all investments in the soma. And so our argument is that we should see life history trade-offs in cancer defenses across the tree of life. I do also want to point out this concept I keep talking about, and I started the talk talking about the number of times a cell divides and getting mutations. All else being equal, the number of mutations that lead to cancer, you need to look at the number of cells that are dividing. So organisms with more cells should have a higher probability. And then also the lifetime of an individual. The longer a cell sits, the more opportunity for that cancer-causing mutation to arise. But if we make some predictions based on life history across the animal kingdom, we suggest that organisms that invest in reproduction will have higher cancer prevalence than species that invest in somatic maintenance over reproduction. And this is interesting because I was traditionally trained studying life history, and I didn't start studying cancer into my postdoc. And what I found in the cancer field was something called Peto's paradox, and I read it and I thought, oh, that's just life history theory, but they just proposed it as a paradox. And sometimes they get mad at me because I'm like, it's not a paradox, that's what life history theory is. But okay, we're going to call it a paradox still. But the paradox is that if, again, all else being equal, a mouse has fewer cells than a human has fewer cells than an elephant. And so if we had all the same cancer defense mechanisms, elephants should get more cancer, right? Because they have more cells, there's just a higher probability of that cancer-causing mutation to occur. But that's not what is observed. And when I say this, this was proposed in the 70s where they didn't have cancer rates in elephants, but they were noticing that not all elephants died of cancer or something like that. So that they proposed that they must have extra cancer defense mechanisms that co-evolve to allow them to be big and long-lived. I should point out a mouse cell, a human cell, and an elephant cell are approximately the same size. So it's not that elephants just have bigger cells. The size of the cell really depends on the function, but also the amount of genomic DNA that's within each cell. And what we've seen come out doing comparative genomics approaches is that elephants do have extra copies of a really important cancer suppressor gene called P53. Humans have have 2 alleles of P53. Elephants have 40. They've duplicated this gene multiple times across the tree of life, or within their genome. Sorry. They also have duplications in a really important gene called LIF. And so this potentially is how elephants have solved the big bodied long life problem. But we still haven't gotten to the data. Do they get cancer? Do bigger and longer lived animals get more cancer? And then the second question I wanted to ask was, do we see trade-offs between cancer and reproduction? So what we did is we contacted colleagues at the San Diego Zoo and the San Diego Safari Park and started working with them on their cancer or their pathology records. So they had a postmortem data set. Typically animals within a zoo setting always get a postmortem. And so that they will, even if cancer wasn't a problem and a cause of death, they can still be uncovered during a necropsy. So we have 42 years worth of data. We have 852 necropsies. This represents 37 mammals and 11 mammalian orders. It's actually quite a small data set. It's some of, at the time, it was the biggest of its kind. I'll talk about the most recent paper, which blows this out of the water by many more species. But at the time, this was all we had, and this was a really neat data set because we were working with a pathologist, and he was diagnosing everything. So it was a really fantastic, highly curated data set. They were able to estimate not only neoplasia, but malignancy, which has been very difficult to kind of get at in some of these comparative approaches. And each species had to have 10 individuals. And again, at this time, this was like the best we could get, which was still small, okay? That's the caveat. And we had quite nice life history data across species. We had elephants in the data set. We had tiny species that reproduce quickly. We had singleton births, and we had opossums that have up to 13 offspring per reproductive bout. And so then we could ask, could life history theory offer an explanation for these cancer defense mechanisms across the tree of life? We did all of our analysis controlling for phylogeny because these data points are not independent because of ancestry. So the data you'll see coming out, all of the regressions I show are taken phylogeny into consideration. And so let's look at the data. What we see here is we're just looking at malignancy, but we've also done it for neoplasia. So we have malignancy prevalence on the y-axis, and on the x-axis, we have our life history trait. For this one, we're looking at body mass. And again, we see this flat line. Bigger species are not getting more cancer in this data set. So we say, yes, this supports Peter's paradox, but it also supports life history theory, right? I'd like to provide those graduate students I had a graduate student run this analysis, and they said, oh, this isn't significant, the model isn't significant. And you're like, no, it's not supposed to be. This is a good thing about this prediction, right? This prediction is there shouldn't be a relationship, and that's still important too. So think about that when you're running these models, and if you expect to see a relationship, or if it's interesting that there's no relationship there, right? I do want to point out there's some species with pretty high malignancies. So opossum, the prairie dog, koalas right here have this known virus. I think it's a papilloma virus that was inducing some of these cancers. Some of you might point out, oh, that Tasmanian devil, they get these facial tumors, these contagious facial tumors. Oh, sorry, I'm getting ahead of myself. Surprisingly, the armadillo and the moose had 0 reports of neoplasia or malignancy. So I think that before this, we really focused on elephants, right? But maybe not. Elephants still get 20% of malignant cancers in this data set, but armadillo and moose had 0% prevalence. So highlighting some new model species. Okay, Tasmanian devils. They get this facial tumor. It's transmissible. It's cancer from another species that's growing tumors on their faces. Just a reminder, this is zoological data, so they don't have the contagious cancer within this zoo setting. They're free from it, but they're still at this higher vulnerability to cancer. And we can chat about why we might think this is still problematic in the Q&A. If we look at lifespan, again, we see no relationship between lifespan and malignancy in these species. And then lastly, looking at litter size. So this is our attempt to look at trade-offs in reproduction. Do species with larger litters get more cancer? Is there a trade-off? And we do see a trend. Again, it's a small data set. I don't want to run around saying that this is indeed biology. There are trade-offs. Cancer is definitely a complex disease, and so it's not likely that all cancers are trading off with reproduction. But this is exciting. We think we can move forward in analyzing a larger data set soon with these results. I also did want to point out that there's another feature of mammals that has been proposed that we are vulnerable, humans are vulnerable to cancer because of our features of our placenta. And so across mammals, the placenta can invade at different levels. So there's some species, if we look at fetal tissue and maternal tissue, have very low levels of invasion. The placenta tissue, the fetal tissue, because the placenta is derived fully from fetal tissue, they don't invade much at all. The cell layers are just touching, and then you have this intermediate form where you're breaking down one tissue layer of the maternal tissue. This is endothelial chorial. These are dog and cat placenta types. And then humans have the most invasive placenta form. And individuals argue the placenta then in the human form shares some of the phenotypes of cancer. It invades, it recruits blood vessels, it induces an immune response, usually evading the maternal immune system. And so sharing these phenotypes of cancer. So it was proposed in the past, and there's some data, some limited data to suggest that cancer malignancy co-varies with the depth of placentation. Again, where the more invasive placenta, these species get more malignant cancer. And that's just a trade-off. We tested this in our data set and we found no relationship. And I was actually really surprised because it was like hitting the two intersections of my work. I study placentas and I study cancer biology. So I thought, yeah, this is why we're most vulnerable. Like we have these super invasive placentas. But then again, this is a limited data set. We just tested it. I don't have the data to show on our larger data set, and we're still not seeing a pattern. We just don't see the pattern. I've been thinking about why. We can talk about it more in the Q&A, but cancer is just an umbrella term. There's actually 100 different diseases of cancer originating from different tissue types. You know, you have carcinomas that are originated from the epithelial layer, to sarcomas, which are more connective tissue, muscle, and bone. And so maybe we're being too blunt with our analyses right now, and it's fine. This was the first step, but now we need to start refining our tool set. And we actually need more data, and we need better data. And this is a story of potentially me being quite naive when I started this project, because I started contacting zoos to get more data and more better data because of the arrow pointing towards human health and disease. And I said, oh, I'm studying cancer in humans, and I want to know if your animals get cancer, and do you have data on it? And they're like, oh yeah, we do. And then I'm like, can I have all of it? And they said no, because that was a lot of work and I was all over the place. But also I naively didn't recognize that there were many species in managed populations that were being diagnosed and dying from cancer all the time as well. And it is a One Health problem. And since working with the zoos for the last 10 years, I've really taken this One Health approach, and animal patients are patients as well, and working more cohesively with our veterinarian colleagues as equal contributors to asking this question has really helped our team move forward. So that being said, we started working with a larger database in collaboration with a French team. And so they had access to this database called Species 360. And so this has almost all the managed zoo population, at least within the US, but also internationally, many of the zoos, they have husbandry and health records in this data set. So now we've gone up from 36 species to 191 mammal species. So it's a much larger data set. And then we're looking at cancer being registered only if it was the cause of mortality. So we're really looking at cancer mortality here in this data set. And we can ask the same questions. Do body size and lifespan trade off with cancer? And so what we see in our cancer mortality risk across species is that, again, there's no phylogenetic patterns potentially other than carnivora. And so we do show in this paper that the meat eaters, especially mammals eating mammals, have higher cancer prevalence, and we don't know why. I don't think it's diet per se, like as the individuals, what they're eating. There could be constraints of being a carnivore. These species, some of the larger carnivores, have gone through genetic bottlenecks as well. And so it's probably a multifaceted explanation. We do confirm that these moose are pretty cancer robust, at least in our data sets. It's an interesting species. The elephants really are always on the front side of things, but maybe it's the moose in the background that's a species we should be studying. And so we retested in this much larger data set if there was a relationship between body mass and lifespan. This is now age-corrected data too, because cancer is a disease of aging populations. We have ages at death for these species, and so we were able to do age adjustments. And again, this is robust. There's no support for Peto's paradox. Bigger, longer-lived animals do not get more cancer. Okay, so and this paper is published just in 2021. We have had some, this is again just zoo data, right? And so when we're talking about evolutionary mismatch, anything like this, we get some criticism that this is an unnatural population we're studying because they're managed in facilities. We have had the opportunity, this is where maybe our story took an unexpected turn. I wasn't expecting this, but Kenya Wildlife Services reached out to us and said, we see tumors in some of our wildlife animals. Will you come start collecting data on this? And so we took a trip out there. And they have a mobile vet unit that surveys the wildlife, especially the bigger wildlife under their care. And mostly it's for poaching and infectious disease. But what they do see, in the next slide, I will show an animal tumor right here, that, yeah, we see wild animals getting cancer as well. It's much harder to quantify. We have limited data. We don't have denominators. We can't really calculate a prevalence, but we have case studies. This is a tumor in an elephant. Again, the data is limited because we can only see these external tumors with this wildlife survey. But what we do see is tumor in the elephants and these sarcoids in zebra populations. And so we will be following up with this group, trying to see if there's at least some mechanisms of the same types of cancer we could be seeing in wildlife versus managed populations. And this has also led us on an unexpected journey to studying cancer across many different organisms other than mammals too. And so crocodiles is a big project for us. This is one of our vet colleagues dissecting an elephant tumor in Kenya. And then snakes actually get quite a high prevalence to cancer as well. We're currently looking outside of mammals, and so we're working with the Exotic Species Cancer Research Alliance, again, building a larger data set. And so some of our next steps, we're going to be looking at cancer across reptiles and birds and amphibians, but this is just preliminary data. And what we're seeing in signals on this data set time and time again is that mammals are more vulnerable. And we're unsure why. We're unsure if it's because mammals are furry and cute and get more recognition, or if there indeed is something different about their biology. We can talk about this again in the Q&A, but I do think there's something inherently different about our biology, and that's being a placenta mammal and having to have immune tolerance beyond yourself. And maybe that does lead to a trade-off or a consequence to being more vulnerable to cancer. Okay, I know I only have potentially a few more minutes here. I'm still good on time. Okay, good. So I'm going to transition to say, you know, we looked at life history trade-offs across the tree of life. Does this framework apply within human populations? Can we see the same trade-offs? And for this, we're going to use breast cancer as an example. I'm inherently interested in breast cancer because it's one of the top causes of death in US women. It's a leading cause of cancer globally within women. And it's also really interesting from an evolutionary perspective because we have seen shifting patterns in reproduction. We've seen humans that go through a transition and become more living in a more industrialized society, have suddenly a later age at first birth. You might have fewer pregnancies, and you see this increase in cycling and menstrual cycles in women living in industrialized populations. And it's been suggested in the past that breast cancer is associated with this higher cycling. It's estimated that individuals living in hunter-gatherer type populations and natural fertility populations have about 100 menstrual cycles in their lifetime. That's compared to individuals in industrialized populations that are non-natural fertility populations have about 400 menstrual cycles in their lifetime, so substantially more. We also see, so that's maybe more of the evolutionary mismatch component of it, but also within breast cancer, there's some suggestion of antagonistic pleiotropy as well. And this is the example, BRCA. So this is a tumor suppressor gene. Mutations in this gene increase an individual's risk for breast cancer. However, it's been shown within one study that BRCA has implications for human fertility. So it's one of these genes that play this dual role between fertility and DNA repair. So individuals with BRCA mutation, again, have a 40 to 80% lifetime risk of developing breast cancer. But there was a study in 2011 that showed that this BRCA mutation, these individuals have a shorter interbirth interval and more children. So it could be one of these antagonistic pleiotropy trade-offs that we're seeing within this. So when I started this project, we thought, well, let's back up and think about it in a life history framework. Can life history help us think about breast cancer along with these other evolutionary perspectives? And so we asked the question, do humans invest more? Do some humans have higher somatic maintenance? And if so, should you see that trade off with reproduction? Okay. And again, as we see that breast cancer is often characterized as a disease of reproductive mismatch, and that's it. So like I'm begging for like a more life history component to this, and it's not just evolutionary mismatch. So typically what you see in some of these studies is that late age at first birth and low parity are risk factors. And the argument is it's because this increased cycling. And we don't know much about early life factors and how they could lead to later breast cancer susceptibility. I suggest that maybe there's some complex ways to shape cancer risk, and that early life and reproductive life are an important contribution, all right? So in this study, we specifically want to focus on early life environment and how that could shape disease risk later in life, but also looking at that reproductive period because we know the reproductive period is really important. So looking at early life, the reproductive period and outcomes, and then breast cancer. And we, in general, if you look in the literature, early life and cancer risk later is slightly correlated. There's studies that look at childhood dysfunction. They look at adverse childhood experiences, the ACE measures. These individuals have just all-cause mortality, more all-cause mortality. There's some studies looking at childhood adversity and the increased risk of lung cancer, premature death from lung cancer, and this is controlled for by smoking. It's suggesting maybe other possible mechanisms. And then there's an association between child physical abuse and cancer, again, just one paper. So there's very few papers out there looking at this early life setup in cancer risk later in life. Although we argue that, you know, just calibration of somatic maintenance could be different from this early life setup. And so with our data set, we wanted to say individuals in these unstable, unpredictable early life harsh environments will have lower cancer defenses, and then they'll have higher cancer later on in life. In order to test this, we started working with the Utah Population Database. This is my colleague, Ken Smith. He's the director of the Utah Population Database. This was him saying how wonderful the database is to Joe Biden when he did his cancer moonshot years and years ago. But those of you that are not familiar with the Utah Population Database, it has an extensive family histories of Utah. They're linked to their demographic and medical information. There's about 7.3 million individuals, and it's a historic database. It starts in the 1880s and goes to the present. So you can look at this intergenerational stuff within this database. So what did we do? We took the Utah population database and we took women. We had it matched with the Utah Cancer Registry. So we had data from women diagnosed with cancer from 1990 to 2010. We had 25,000 cases, and then we just age matched the controls. What we then did was develop a novel adversity score. It's different than the ACE measures because we're looking at demographic data. So we call this a more structural data set. We're looking at income. Did your parent die before you turned 10? Like these early life traumas. So it's motivated by the ACE experience, but it doesn't suffer from recall bias because it's more structural stuff that we're pulling from the demographic records. And so we have 6 measures. We call it a cumulative adversity score. If you have one of these, your score can be from 0 to 6 in your adversity score. Each knock gives you a point. And so you could have premature death of your mother, your father. You could have siblings that died in childhood. You can have SES. And then we have an income to need ratio, the number of children in a household, along with low SES. And this is our cumulative adversity score. And so the first thing we did is say, hey, does this check out? Is it operating the same way we would predict it to? We do see individuals with higher adversity have an early age of death. I see the blinking. I'm almost wrapping it up, but I wanted to acknowledge that I do see you. We also wanted to validate our cancer study. Does early age at first birth and higher number of children, is this protective against breast cancer? Yes, it is. This is all what we'd expect to see in this data set. So then we asked the question, does adversity predict breast cancer, right? And the answer is no. We didn't find that. Okay, so I thought it would be a cumulative effect where you'd see, like, if you're really worse off, having cancer would just push it to farther, right? We did not see this. We also looked at an interaction. We found no significant interaction as well. And so it's interesting because these women are dying earlier with our adversity score. It's just not of breast cancer. Why? Well, what we do see is individuals with high adversity accelerate their reproductive scheduling. They have an earlier age of first birth. And this is consistent with life history predictions as well, right? So it's complicated over the life course. We've also looked at these individuals with high adversity actually have a later age of diagnosis. Again, this could be due to lack of screening because low SES is a high adversity measure, and we know that individuals living in low SES environments lack medical care compared to high SES environments. Or it could be because high adversity leads to an earlier age at first birth, mediating this, which just ends up leading to protective against breast cancer, right? Um, and in this, I'll just finish right here. So maybe early age at first birth, how can it influence breast cancer risk? Well, I talked about the cycling. The age at first birth maybe introduces fewer menstrual cycles. I'm not really convinced by that. I want more than that because parity didn't have an effect here. It's the age at first birth. It's the timing. There's another suggestion in the literature that age at first birth is protective against breast cancer because the differentiation of stem cells in the breast tissue. And so the earlier those get differentiated, the less cancer-causing mutations that can arise. And so those are the competing explanations why. And the answer, unfortunately, right now is we don't know. But it's not what I expected it to be. The other thing I want to just end on is breast cancer. I'm saying again, is this like one disease situation, but it's not. It's heterogeneous. There's many different kinds of breast cancer. A lot have to do with hormone status. So actually in our follow-up, we're going to look at this, and we should be able to tease apart the menstrual cycling because that should be estrogen receptor positive status. And so once we start controlling for actual hormone positive or negative breast cancer, maybe we'll see a effect then, and we're predicting that we will. Okay, so that's it. I want to have time for questions. Cancer, I started with the talk talking about it being a part of multicellular organisms. We know that species vary, and it's due to various evolutionary constraints and trade-offs, not just one mechanism. We see complex interactions happening here. There's some support that humans are more vulnerable, potentially due to evolutionary mismatch. However, it may be a shift in cancer type, especially when we're thinking about breast cancer. I don't think the menstruation story is the only story to tell here. And that breast cancer is a heterogeneous disease, and we have different life history components that we should be thinking about with this heterogeneous disease. And with that, these are my acknowledgements. A huge team. I work with the Huntsman Cancer Institute. I work close with ACE team members at ASU and our veterinarians.